Good morning, friends. Myself, Dr. P. Samuel, Assistant Professor of Biotechnology of Ayya, Nadar, Janaki, Ammal College, Sivakasi. So, through this video tutorial, we are going to discuss an important topic in bioinformatics, and the topic is called sequencing and sequence submission. Right? So, this is an hardcore topic in the field of bioinformatics. Most of us used to study what is sequencing, what is sequence, and what is sequence submission, everything. But you should understand some basics and the requirements for sequencing, as well as how to submit a sequence in a proper way. Right? So let's move on to the content now. So here I have given two pictures. The picture what you are witnessing on the right hand side, on the left hand side, is the sampling spot. Right? This is the sampling spot image, which is located across the southeast coast of Tamilnadu, and the seashore is richly contain sea grasses. Right? You could find a huge population of sea grass just below the surface of the water. So the sea grass used to grow here, and they develop, they promote their colonies, and they eventually die in the same place. As a result, the soil of this particular locality is looking black in color. See, on the right hand side you are viewing the image of the soil collected from the sampling site. See how the soil is looking like. It is very, very black in color. The black nature of the soil is due to rich carbon content. Right? So this is a very good sample for microbiological examination. So you should collect the sample in an aseptic way, so that there will not be any intrusion for the entry of organisms from our side. Right? So you should collect the sample whenever you are collecting the sample, and wherever you are going and collecting the sample, collect the sample in an aseptic pattern. This is very, very important in microbiological analysis. Once the sample is collected, it is transported to the laboratory, where we are using a media. Here I am giving an example called fungus. So for fungus I am using a medium called PDA, which means potato dextrose agar medium, and this contain dextrose as the source of carbon, which is easily utilized by the fungus for its growth and establishment. See, here you could find different types of colonies here, right? Fungus 1, fungus 2, fungus 3, right? So I am going to identify these fungus, right? So this is very base in research. This is the fundamental step in research. So whenever we are stepping into microbiological research, the first step is to identify the organism. See, for identification purpose, for fungus we are using a most common procedure called microscopic identification, and this identification is not given priority in the field of research. But what people are seeking is to identify the fungus based upon its molecular makeup, which means molecular level. Molecular level identification is asked by the people across the globe. So for that purpose, what we are doing is, we are going to isolate the DNA from the fungus, and we are going to sequence it in order to identify what fungi is growing in your Petri plate. Right? So for that purpose, what I am doing is, I am extracting the DNA. Right? So the most common method for for isolation of DNA from fungus is CTAB method. Using CTAB method, I am extracting the DNA from the fungus. Right? So after isolation of the DNA from the fungus, it should be purified. See, why it needs to be purified? Because after the isolation procedure, along with DNA, there are many contaminants. See, for your easy representation, I am giving black color dots into the coiled structure. See, just think that these black color dots are impurities, which may be proteins or which may be RNA molecules, whatever it is, right? So it is your responsibility to purify the DNA through sequential steps, right? By centrifugation process, right? So through sequential procedure, we are removing all other impurities so as to obtain pure form of DNA, right? Our aim is to obtain pure form of DNA, which in turn used by the people for sequencing studies, right? So once the pure form of DNA is obtained, we are going to run a procedure called sequencing, right? What is sequencing? The ultimate question arises in your mind is, what is sequencing? See, sequencing is a procedure to identify the nature and the position of nucleotide in the DNA, right? So sequencing, I repeat, sequencing is a procedure through which we can identify the nature and position of nucleotide in the DNA or RNA. So it is called a sequencing procedure. Once the sequencing procedure is completed, you supposed to obtain a result. The result is called a sequence. The result is called sequence. So sequencing is a procedure. Sequence is the result of sequencing. So at the end of sequencing, you will obtain a long stretch of nucleotide arrangement data. So this could tell you the nature and position of nucleotide in the DNA, what we have provided into the sequencer machine for sequencing purpose. See, two things. What you need is a biological organism which is acting as the source of DNA. So from the organism, you have to isolate the DNA by adapting the suitable procedure. After isolation of DNA, the DNA is purified. After purification, it is sequenced. After the sequencing procedure, you have identified the nucleotide composition of the DNA sequence, right? So we'll come to the end of the session. So after obtaining the DNA molecule, we should sequence it, right? So thank you for watching the tutorial, and we will meet in the second video for the submission of sequence into the database. So stay tuned with me. Bye bye.